Hello, everyone, and welcome to the Day 28 podcast. There is only one objective here, but it's a pretty detailed objective, so I'm going to break this up into two separate podcasts. The first podcast will cover all the way from direct observations of evolutionary change through vestigial structures. The second podcast will cover convergent evolution, fossil records, and biogeography and theory. All right, so let's go ahead and get started with this first pod. There are several examples that we could look at to illustrate how evolution is occurring and still occurring today. There's some nice examples of the myelinase gene that we may talk about in class, or how lactose tolerance is only found in humans. All right, your book talks about an example of the soapberry beetle. And the soapberry beetle is a standard looking kind of beetle. It has what is called a beak, what it feeds with in its head. And the length of that beak is proportional to the type of vegetation that is in that area. We know that the vegetation is different from one region to the other, the beak size will also be different. And a good example of what's happened here to kind of help explain how some beetles have evolved different kinds of adaptations is looking at the state of Florida. We know historically that in the Florida there was a vegetation called balloon vine, and it was the native species, and it grew all over central and southern Florida and a little bit in the north too. And we knew that the soapberry beetle fed on it. In fact, if you looked over history, mainly by looking at museum samples and previously published data, if we made a graph of the beak size, in fact, let's give it some numbers here. We'll use what the book kind of explains it, 6, 7, 8, 9. We know that the beetle that fed on the balloon vine in Florida had an average beak size of around 8.5. Now, because there's variation in this population, we know they weren't all that. We know that they spread all different sizes here. But on average, they were 8.5 millimeters. Now what happened though was in this central region a new species was introduced, and this new species, we'll just draw it like that, is called golden rain tree. The beetles were still feeding. In fact, they were still feeding on this balloon rain here, and this new vegetation which overtook the balloon vine still allowed the golden rain tree still allowed the soapberry beetle to feed on it. Over many, many years, what happened though was that we went back, not we, but scientists went back and looked at the size of the beak of the beetles. And what they discovered was that in these regions here, where there was still balloon vine, the length of the beak did not change. It still had roughly the same kinds of distribution. So I'm going to put a dotted line. And let's make a key here. Straight line here means past beak size, and then the dotted line is current beak size. When they went back and sampled from this region, the region that had 8.5 millimeter beak size previously, they've discovered something different. The average size was somewhere around 7. Still had some range. It was a little bit more skewed this way. Something like this. So the straight green line is beak size with golden rain. So previously, we can see that the beetles in this central region fed on the balloon vine, but when we introduced a new vegetation to it, it shifted, and we see this extreme here. Just to review a term, we could say that this is an example of directional selection. Directional selection because we have shifted all the way towards one side. Okay, so what happened here? How did this shift change? Well, we know that the balloon vine has a much thicker outside, so the beetle requires a longer beak in order to reach the nutrients of this vegetation, whereas the golden rain has a thinner coating, and so it doesn't require as long of a beak. So over time, this shorter beak size was selected for, and the longer beak size was lost because it was no longer needed. Now you still maintain some variation though. All right, so let's review a couple things here. We already reviewed that this was an example of directional selection. But let's also ask this question: where did these alleles come from that allowed us to have this larger beak? And remember, the answer to that is that the alleles weren't generated brand new. These alleles were always in the population. But because we had a change in vegetation, it allowed for the selection of these smaller sized beaks. And one thing we could say about this population that used to look like this as far as the beak distribution, and then shift over here, we could say this is an example of what happens when a population falls out of Hardy-Weinberg equilibrium. When we see evolution within a population, see this population changed here in relation to what it used to be and its neighbors, it fell out of Hardy-Weinberg principle because it violated one of the 5 principles. And the one it changed was this natural selection. We were able to select for a different size beak because of the vegetation. For another example, you'll want to look in your book at figure 2214, and it talks about antibiotic resistance. So let's talk about our next topic, and our next topic is homology. And homology is just similarity resulting from common ancestry. We should also talk about how we can break this down to molecular homology and anatomical homology. Anatomical morphology is sometimes called morphological morphology, but your book uses anatomical, so that's what we'll use here. Molecular homology is just a subcategory of homology. So this similarity resulting from a common ancestry is due to shared molecular similarities. And when we talk about this, we're primarily talking about DNA. Anatomical homology is similarity resulting from a common ancestry due to shared anatomy. Anatomical homology has been the main use of the word homology for many, many years, for decades. But with our understanding of genomes and how to sequence DNA and understand the molecular aspect of organisms, we've been able to support many of our anatomical pieces of evidence with molecular evidence. So first we're going to talk about some anatomical homologies and how we can use that. So let's erase this. So we often think of evolution as this sort of remodeling process, this process of taking what our ancestors had and modifying it based upon certain selective parameters. So if evolution really is this remodeling process, we would expect some related structures between species and their ancestors. Figure 2215 in your book, and we'll show this in a moment here, is a good example. It talks about the 4 limbs of mammals and how they are homologous structures. So let's write homologous structures. And our example will be mammal forelimbs. So in the book they talk about human forelimbs, or human arms. We talk about the cat leg, the whale flipper, and then the bat wing. These all look quite different when you just glance at them, and we know that they have quite a different function as well. human arms, they serve several functions, but let's say they're designed for lifting. Cat legs for running. Whale flipper for swimming. Bat wing so they can fly. With such diverse functions of these forelimbs of these different mammal examples, we might predict that different anatomical structures would be found when we start taking a closer examination of these forelimbs. But what we find is that there's remarkable similarities between them. And now I want to show you the picture in your book of these and go over what is found. Okay, so what we see here in the human arm, the cat leg, the whale flipper, and the bat wing is we see very similar bone structures. We see a humerus in all of them. When you look at them, they're different sizes, but they have the same kind of anatomical landmarks on them. We see a radius and ulna, the two lower bones in our arms. We see those in cats, and we even see them in the whale flipper and in the bat wing. Anatomically, they have very similar structures, though they're different lengths, and in some cases different thicknesses. We also see our wrist bones, the carpals. We see them in each of these diverse animals. We see the bones in our hands. They're called metacarpals, but these are the bones that are in the palm of our hands. We see these in all of these animals as well. Different shapes, but again, anatomically, they have very similar landmarks on them. And then our fingers, the phalanges, we also see those in these other organisms. Again, very similar anatomical features of them, though they have different lengths, and they have adapted so that they can serve a different function. It would be incredibly unlikely that each of these diverse organisms that are common in some ways because they're all mammals, but it would be very unusual that they all four evolved separately and came up with these exact same bone structures. It's more likely that there was a common ancestor that had these bone structures, and then over time, a long time, certain features were adapted for based upon certain things, based upon natural selection. The ability to lift, the ability to run fast, the ability to swim, the ability to fly. These are all natural selection processes that would have selected for, over time, these features. It's important to remember you didn't go from not having a flipper to having a flipper. It was a very slow process where you went from a functional forelimb to it becoming less functional, less functional, less functional, until it adapted into a functional flipper. Again, this is not an overnight thing. This is many years, many generations that this would take. All right, as I hinted to earlier, these anatomical structures were discovered by biologists a long time ago, and then recently, say within the past decade or two, this has all been supported by molecular homology. The genes, the developmental genes that lead to an arm, to a leg, to a flipper, or a wing, are remarkably similar between these four different organisms. And again, it's not a single gene, it's a whole set of genes. But these genes have very few nucleotide differences from one organism to the next. But those changes are significant because they do change the actual function and the structure of these different adaptive modifications. So again, anatomical homology has been supported by molecular homology. The next thing we want to look at is something we'll call developmental. When we look at adult organisms of different species, say looking at a chicken versus a human, those look quite different. And it's hard sometimes to look at those just on the outside and say, how are they similar? And we talked about how we could look at their bone structure, we could look at their organs and see they are actually quite similar. But there are some features of birds and fish that mammals don't have. Often, however, when we go back and look at the embryonic development of an organism, we can see some of those shared features. So for instance, all vertebrates, as an embryo, they have several things in common that we don't see in all vertebrate animals. So as an embryo, the developing embryo, they all have what are known as pharyngeal arches, and they all have a postanal tail, and many other structures that we see in the embryo. Let's go back and look in your book real quick at these two embryos. We see on this side a chicken embryo, and here we see a human embryo. They have some very similar features to them, but we see these two things that we want to point out. The pharyngeal arches, we can see right here in the head region, but below the head. And then over here we see it in the human embryo as well. We also see this postanal tail here in this chicken embryo. And then we see the post-embryo tail here in the human embryo. We can see by looking at these embryos that there's a lot of shared homology, shared anatomical homology within these embryos. So what happens as this chicken develops into an adult chicken, and how this human embryo develops into a human embryo? To these pharyngeal arches and the postanal tail. The postanal tail in the chicken persists as a tail as the adult. In the humans, as you are probably well aware, we lose this tail. It slowly regresses, and it regresses, if you remember the term apoptosis that we introduced when we talked about signal transduction, the programmed cell death. That's what happens here. These cells are programmed to die, and slowly that tail slowly goes away. Interestingly, every now and then a human is born with a very small kind of vestigial tail, and usually the doctor just chops it right off. Doesn't happen often. Okay, now what about these pharyngeal arches? Well, in humans, these go on to become part of the ear and the throat, that pharynx region in our neck. Similar structures form in the chicken, but things like a fish, they go on to form gills. So both humans and fish have a very similar structure that in fish becomes gills, and in humans becomes part of our throat area. All right, let's move on to this next topic of vestigial structures. A vestigial structure is a structure that has little or no known function in the current organism. So we'll write little or no function. But this vestigial structure, we have evidence, did have a function in an ancestor. Your book cites two examples here. They're pretty good ones. And the fact that snakes still have vestigial pelvis and leg bones. They're certainly not used as leg bones or pelvises in their current form as a snake, but their most common ancestors were walking reptiles, and so they did have a purpose for legs and pelvises. But over time, since natural selection acted against these legs and the pelvis in favor of the way snakes currently move, these structures have slowly been losing function and eventually will probably just disappear. The next we see in various kinds of the blind cavefish, they have a remnant eye structure under the skin. So you might have a fish like this, where usually they would have an eye here, but it's kind of buried inside of the fish. It's not a functional eye anymore, of course. It's what's left of the eye. And we know that the blind cavefish, its most recent ancestors, had a functional eye. Or I shouldn't say its most recent, I should say one of its ancestors had a functional eye. Over time, because living in these caves where there's no light and no ability to see, the need for an eye is no longer there. So natural selection has acted against that eye. But we still see remnants of it. Because again, natural selection doesn't work in the way that one day you have a structure and the next day you don't. It takes many, many years for that structure to either go away or to be adapted upon to become a more functional structure. If evolution occurred in such a way that these structures formed independently, not from a previous ancestor, it would seem a little odd to have a remnant eye structure in these blind cavefish or these vestigial leg bones and pelvises in snakes. There would be no reason to have them if it wasn't from an ancestor. All of these examples of the vestigial organs, the homologous anatomical structures, the embryonic developmental evidences, these are all anatomical structures, morphological homology pieces of evidence. All of this has been supported in the past couple of decades with molecular evidence, showing that the genes for eyes, the genes for a leg bone and pelvis, have also changed as these organisms have adapted to their new environments. All right, well, that ends this first podcast on the first part of Objective 1. Watch the second podcast to see the rest of this objective. We'll see you in class. Bye.